Yes, sir, clear.
Last class we completed in cell biology topic, cell wall completed, plasma membrane completed, and nucleus completed. What you learned in cell wall, plasma membrane, nucleus, just give me in brief answer. Cell wall, what you learned about cell wall structure, Jyoti,
Yes, sir. Cell wall provides the support
air support,
and also
okay,
it it's the protoplasm which gives protection from external injuries.
I'm asking cell wall structure. Cell wall made up of how many layers?
Uh, Three layers, sir.
Right, three layers. Primary, first
Secondary and
one, first one middle layer, primary layer, and secondary. Secondary layer only developed in well-developed cells. Right. So the chemical modifications, middle layer magnesium, calcium pectate, primary cellulose, secondary pectin, suberin. So that is a chemical. Plasma membrane, phospholipids and proteins, and also we learnt about the models also, fluid mosaic model, and we discussed about nucleus too, right? So what are present in nucleus? What are present in nucleus? What are the components of nucleus? Nuclear membrane, nucleolus, chromatin thread. These are the major portions. What is present in chromatin thread? How the made up of chromatin? Hello,
it consists of DNA and proteins.
right? Consist of DNA and protein. Now today topic is about the DNA structure. So this is the order. Okay. So in last class we discussed in cell part, cell component, cell wall completed, plasma membrane completed, nucleus completed, and in nucleus mainly the major hereditary component chromatin. Chromatin, as you said, it is made up of protein and DNA. So now today topic is DNA structure. Let's see the DNA structure. Just wait. Are you able to see this one? Hello?
Yes, sir.
Hello? Right. DNA structure. What is DNA? What is DNA? What you learned so far about the DNA? Here the nucleic acids are two, DNA and RNA. First, let's discuss about the DNA. Next, followed by RNA. Now tell me the DNA. What is full form of DNA?
Deoxyribonucleic acid.
Right, deoxyribonucleic acid. Let's see the structural components of DNA. DNA mainly structurally made up of nucleotides here. Nucleotide, nucleotide chain. This nucleotide made up of nucleoside. Nucleoside. The nucleoside made up of nitrogen base and sugars. So mainly in DNA, main major components are phosphoric acid, pentose sugar, and nitrogen bases. Here these three, phosphoric acids, pentose sugar, nitrogen bases, these three together form as a nucleotide. Nucleotide. Here this one phosphoric, this one phosphoric acid, and this is sugar, and this is nitrogen base. This all together formed as a nucleotide. Just wait. this is phosphoric acid. Okay, this is sugar, sugar, ribose sugar, deoxyribose sugar. This is nitrogen base, nitrogen base. So this is one nucleotide. This is one nucleotide, right? So next, sugar, phosphoric acid, same phosphoric acid, sugar, phosphoric acid again, again sugar, again nitrogen base, nitrogen base. This is one nucleotide. So one nucleotide, second nucleotide, like this number of nucleotides form. Number of nucleotide form together as a chain, like this chain. Number of nucleotide form together chain. That's why it is polynucleotide. This is called polynucleotide. So each nucleotide made up of nucleoside means nucleotides, nucleoside and phosphoric acid. This is phosphoric acid and already told you nucleoside made up of nitrogen base and sugar, sugar and nitrogen base. These two, except phosphoric acid, let's change color, blue color, except phosphoric acid, sugar and nitrogen bases called as nucleoside. This is nucleoside and this total complete red color nucleotide, right? Is it clear? Hello? Hello?
Yes, sir.
Right. So now let's discuss one by one. First, let me complete about nitrogen bases. Nitrogen bases. What are the nitrogen bases? Here the nitrogen bases two types. Nitrogen bases are two types. The two types nitrogen bases are purines, purine, purines, and pyrimidines. Purines and pyrimidines. Two types, purines and pyrimidines. What are the purines? Do you have book?
Yes,
Do you
sir, we
have
are
book?
having.
Right.
Yes,
What
sir.
are the purines?
Purines are the two rings structures. One contains a six-membered pyrimidine ring of benzene and a five-membered
No,
imido.
I am asking no need details. Just what are purine? Actually, the nitrogen bases adenine, thymine, guanine, cytosine. In this, the purines are only adenine and guanine. Adenine and guanines are purine, right? And pyrimidine, TC, TC pyrimidine, TC pyrimidine, thymine, cytosine, pyrimidines, right? So nitrogen bases for, remember the nitrogen bases are four, which containing mainly nitrogens components. That's why these are called and basic nature. That's why called as nitrogen base. The nitrogen bases in DNA four types. These four nitrogen bases, they are adenine, guanine, thymine, cytosine. The majorly these are made up of the chain, made up of the ring. Based on the made up of the rings, it is divided into two types. That is purines and pyrimidines. Purines and pyrimidines. The purines are adenine, guanine, and pyrimidines are thymine, cytosine. Here let's discuss about purines. It may be adenine and guanine. So this both either purine or pyrimidine, sorry, this adenine and guanine, anyone, it is dicyclic, made up of dicyclic. Whereas in pyrimidines, made up of one cyclic. We can see in diagram also. You can see in diagram also. This is adenine. Are you able to see? Hello?
Yes, sir.
Hello? Right.
Yes,
This
sir.
is how many rings in adenine here? How many rings?
Two rings.
Two rings. One is benzene ring, one is benzene ring, one is imidazole ring. Benzene ring and imidazole ring. Two rings. In adenine. At the same way, here guanine also, we can see guanine also. This is guanine. Guanine here also same. One is benzene ring, one is imidazole ring. So it is made up of two rings. Two rings. So these two rings called as purines, adenine and guanine. Whereas in pyrimidine, let's see here, this is cytosine, right? Cytosine, cytosine. Cytosine, how many ring here? Cytosine. This
One
is
ring, sir.
ribose sugar. It is another. This is not a This is not a nitrogen base. It is the nitrogen base. Only one benzene ring. Only one benzene ring. Cytosine. Right. Here also, how many rings here? Thymine. This is thymine. It's thymine. How many rings in thymine?
One ring.
Only one ring. That is benzene ring. Only one ring. That is
Benzene.
benzene ring. Right.
Yes, sir.
So nitrogen base complete. Next one. Let's wait 1 minute.
Sir, what about uracil?
Uracil, actually, uracil present in RNA only, not DNA.
Okay,
Hello?
sir. Okay,
It is
sir. Okay.
DNA structure, but the same structure carry forward in RNA. The only one difference in instead of cytosine, sorry, just wait. Adenine bond with thymine, guanine, cytosine. In place of thymine, uracil present in RNA. Okay? Hello?
Okay, sir. Okay, sir.
Right. So already we completed purines and pyrimidines. Now next one, sugar, deoxyribose sugar. So nitrogen bases computed. Any doubt in nitrogen bases? First of all, before entering this topic, first you should know about the DNA structure, DNA structural components. In DNA structural components, nucleotide. Nucleotides means nitrogen base, sugar, P for phosphoric acid. So one component clear, completed. The one component mean nitrogen base component. Nitrogen base component completed. Is it clear? Any doubt in this nitrogen bases?
No, sir.
Right. Further details we should complete in DNA structure, Watson Crick model. Right. Next one, the sugar. Deoxyribose sugar. Deoxyribose sugar. Here you go. Deoxyribose sugar. Deoxyribose sugar. Deoxyribose sugar. Deoxyribose sugar. Right. So it is present furanose ring form. Furanose ring form. The furanose ring form contain 5 carbons, 5 carbons, 5 carbons, and 10 hydrogen, 5 carbons, 10 hydrogen, 4 oxygen, 4, sorry, 4 oxygen, 4 oxygen. The second carbon atom of the ring is divide oxygen. That's why it's called as a deoxy. So deoxyribose contain, instead of oxygen, OH atoms at presence of third position and fifth position, third position and fifth position. At third position of the carbon and fifth position of carbon, it is adding OH hydroxy group, so that it can form a bond with phosphoric acid. So the third place, bond formation phosphoric acid. Here also fifth place, bond formation phosphoric acid. Here the how many phosphoric acid attached here? For one sugar molecules, one pentose sugar, how many phosphoric acid? Two. One phosphoric acid, third position, another phosphoric acid, fifth position, right? Hello?
Sir, we can't see the screen, sir.
Now able to see,
Yes, sir. We
right?
can't able to see the that deoxy ring.
I'm continuing with the previous image.
Okay,
Are
okay.
you able to see previous image?
Yes, sir. DNA image, yes, sir.
Yes, DNA in DNA image for sugar. Are you able to see this
Uh,
marker?
no. Yes, sir. Marker we are able to see.
Right. Are you able to see the sugar name? Sugar.
Pentose sugar. Yes, sir.
Yes, pentose sugar. This is pentose sugar, pentose sugar, pentose sugar, third carbon, fifth carbon. Here, third carbon, third carbon, phosphoric acid. Are you able to see right mark?
Yes, sir. Yes, sir.
This is third carbon, phosphoric acid. And this is fifth carbon, phosphoric acid. So in middle phosphate. So here we can see sugar, sugar, right?
Yes, sir.
For example, this third carbon, fifth carbon. Third carbon with phosphoric acid, fifth position also with phosphoric acid, right?
Yes, sir.
As for sugar, already told you made up of five carbons. In five carbon, third carbon, third carbon, and fifth carbon, third and fifth carbon attached with OH hydroxy group. Here hydroxy group, here also hydroxy. These hydroxy group form a bond with phosphoric acid, with ester, with ester. So how many esters here formed? How many esters? Here one ester bond, here one ester bond. So it is called phosphodiester bonds. The formation phosphodiester bond. Okay? Hello?
Okay, sir. Okay, sir.
Any doubt in this or confusing or not?
Somewhat confusing, sir. But once we go through it after that.
Oh, no, you don't worry. First, are you clear with DNA structure?
Yes, sir.
Right. You clear with the nitrogen base?
Yes, sir. Nitrogen bases are clear.
Clear with sugar form? Sugar, sugar.
Uh, no, sir.
Simple. Here the sugar made up of, what is the formula of sugar? Deoxyribo sugar formula C5H10O4. Right? How many carbons in this sugar? How many carbons?
Five.
Five carbons.
Five. Yes, sir.
Right. For example, this is sugar. Let's change color here. CH. OH. H. H. Here hydrogen. Here OH. CH2. C. Here hydrogen. This is pentose sugar formula. Okay. Hello.
Okay, sir. Okay.
Pentose sugar formula. In this first carbon, this is second carbon, this is third carbon. Okay.
Okay, sir.
This is fourth carbon. Here fifth carbon. Right? Clear?
Yes, sir. Yes,
So
sir.
here hydroxy group at let's change color. Are you able to see yellow color?
Yes, sir.
Third carbon and fifth carbon. Okay?
Yes, sir.
Clear? The yellow color. This is third OH group. Fifth OH group. So here the phosphoric acid formation. This is phosphoric acid. This is phosphoric acid here. Are you able to
Okay, okay.
see? Right.
Yes, sir.
So the phosphoric acid, let's just wait, let's change color. P, bond formation with third carbon. P, P for phosphoric acid, bond formation with fifth carbon,
Yes, sir.
right? So
Yes.
this is P for phosphoric acid. Phosphoric acid attached to this sugar at third position and
Fifth
fifth,
position.
right, that's it. Third and fifth position. The third position, one ester bond. Fifth position, one ester bond. So it's a bond with diesters. Bond with diesters. So this is sugars bond with P for phosphoric acid. Already told you this nitrogen base, sugar, phosphoric acid all together called as a nucleotide. Is it clear? Any doubt?
No, sir.
Right. Next. Let's see Watson-Crick model. Are you able to see?
Yes, sir.
Sorry. Right. Watson-Crick model, very, very important. Watson and Crick model. If you understand DNA structure, it will be easy to you. First of all, are you clear with DNA structure?
Yes, sir.
Right. So let's see the Watson-Crick model. DNA usually exhibit double helix model, double helix like this, double helix, double helix model, which was discovered by J.D. Watson and F.H.C. Crick in 1953, for which they were awarded a Nobel Prize in 1962. Their discovery was based on earlier investigation by Erwin Chargaff and Franklin. According to this model, that means double helix model, DNA is composed into two polynucleotide strands. So this is one polynucleotide strand and this is another polynucleotide chain. So your polynucleotide chain, sorry, polynucleotide chain made up of what are present in polynucleotide chain? Hello?
Phosphate.
Right, phosphate. This phosphate attached with
Uh,
right as you said phosphate and sugar,
Yes, sugar.
and again phosphate. This sugar attached to the sugar. So we can see here. This is phosphate, right?
Yes, sir.
Phosphate. This is also phosphate. Two phosphoric acid attached with this is S for sugar, right?
Yes,
What
sir.
is this? This A for
A adenine, sir.
adenine. Adenine means nitrogen base. So already
Yes.
told you these three nitrogen base, phosphoric acid, sugar. These three called as nucleotide, right? One nucleotide. This is two nucleotide, three nucleotide. Like this number of nucleotide present throughout the entire DNA strand. Likewise, phosphoric acid, sugar, phosphoric acid, and same again sugar, phosphoric acid, sugar, phosphoric acid, sugar, phosphoric acid, like this chain. Phosphoric acid, sugar, phosphoric acid, sugar, phosphoric acid, sugar, like a backbone, polynucleotide like a backbone, sugar, phosphoric acid. So each sugar, 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 sugar. The sugar bond with nitrogen base,
Sugar.
maybe
Yes,
adenine,
sir.
maybe guanine,
Guanine.
maybe
Yes.
cytosine,
Sir.
thymine,
I mean,
right? This is one end only. This is one strand polynucleotide. This is only one strand structure. Only one strand structure, right? Clear? And next,
yes, sir.
according to double helix model, DNA composed polynucleotide. How many polynucleotides? Two polynucleotide strand, which are spirally coiled. This is spiral, a shape, spirally coiled around the another. These two strands are parallel or anti-parallel. This is always anti-parallel. Anti-parallel means one is 3 and 2, another one is 5 and. One is 3 and another one 5 and. So this is 3 and to 5 and. That means simple. For example, this is third carbon phosphoric acid. Here, fifth carbon phosphoric acid. So this is called 3 and to 5 and like this. For example, one strand 3 and 5 and 5 and. Right? Another one is anti-parallel means opposite direction. 5 and to 3 and reverse. 5 and to 3 and 3 and to 5 and 5 and to 3 and. Is it clear? Hello?
Yes, sir. Yes, sir.
Any doubt in this?
No, sir.
Right. So is anti-parallel to each other and both the strands are held together by hydrogen bonds. Here, here we learnt only one hand. This is only one hand. One polynucleotide. One polynucleotide. Here anti-parallel, another polynucleotide. Another polynucleotide. What is between two? These are what are these? Hello?
Yes, sir.
What are these?
Hydrogen bonds, sir.
Right, these are hydrogen bonds. These are hydrogen bonds. Right? It is attached to hydrogen bonds. Hydrogen bonds. Together hydrogen bond between one purine, one purine is one strand and pyrimidine in opposite strand. Always purine bond with pyrimidine. What are the purines? Hello?
The
Yes,
nitrogen
sir.
such as adenine and guanine.
Adenine,
Adenine and guanine. Yes.
adenine and guanine are the purine. What are the pyrimidine?
Thiamine and cytosine.
Thymine and cytosine. So remember, always purine bond with hydrogen, thymine. Pyrimidine, sorry, purine with pyrimidine. Pyrimidine with purine. For example, here adenine is the purine, thymine with pyrimidine. Is it clear? And another important thing is here adenine is bonded to thymine by two hydrogen bonds, only two hydrogen bonds. Whereas guanine bonded with cytosine with three hydrogen bonds. And cytosine with guanine, three hydrogen bonds, thymine with adenine, two hydrogen bonds, like this. So we can see in diagram also. In diagram here adenine, right? Here adenine. This is thymine. How many hydrogen bonds in between? This is one, this is two, right? Hello, are you able to see?
No,
Hello,
sir.
it's not clear.
Yes, sir. It's not clear.
Just wait. Right now clear?
Yes, sir. It's clear.
1 minute. Just a minute. Sorry for the disturbance. Right? Now clear? Hello?
Yes, sir. Yes, sir.
Right.
It's clear.
How many bonds between adenine and thymine? You can see this diagram. Are you able to see arrow mark?
Yes, sir.
Here, how many bonds? How many hydrogen bond? How many hydrogen bond able to see here?
One. Yes, sir. One only.
This is
One bond, sir.
one.
Hydrogen.
This is one bond, and this is second bond. Two. One and two. One, two.
Okay.
One. Okay. Two. What about cytosine and guanine?
3.
Three, sir.
One, two, three. And thymine and adenine?
One.
2.
Two. One
Oh,
and
sorry.
two.
Three, three, sir.
And guanine and cytosine?
Three, sir.
Three. Right. So here the purine always bond with pyrimidine. Pyrimidine always bond with purine. But here the one important thing is here adenine, thymine, two hydrogen bond, as well same thymine adenine also two hydrogen bonds. But guanine cytosine guanine, three hydrogen bonds. Three hydrogen bonds. Is it clear? Hello?
Yes, sir.
Right. The amount of adenine always equal to thymine. Remember it. Amount of adenine always equal to thymine and amount of guanine is equal to cytosine. This base ratio may vary from species to species, but base ratio different of species to species different, but it is constant always 1:1 ratio. That means according to Erwin Chargaff, purine and pyrimidine exist in 1:1 ratio. Purine pyrimidine ratio 1:1. For example, 10 adenine present in a stand. How many thymines are present? Hello?
10.
That's it. 40 cytosine present in a strand. How many guanine present? 40 cytosine. What about guanine?
40, sir.
40. That is 1:1 ratio. Remember, according to Erwin Chargaff, 1:1 ratio. Always the purine pyrimidine 1:1 ratio. Deposited with 1:1 ratio. Is it clear? Hello?
Yes, sir, it's clear.
Right. And the diameter between the two strands means here, just wait. This is between two strand diameter, this length. The diameter between two strand 20 angstroms. 20 angstroms. 20 angstroms, right? The length of DNA depend upon the number of nucleotides. That's so different. The two strands of DNA are coiled to form a twisted ladder. Twist, like a ladder, but it is twisted ladder. The coiling occurs at angle 360 degree. Turns it here that the spiral ring of DNA turns at 360 degree. Turns at 360. Full turn 360. This coiling leads the formation of a turn. It is complete one turn, or it is called as helical. Each turn of DNA helix include each turn. Each turn include one is minor groove, this is major groove, and one is major groove. This is major groove. So this is called one turn. One minor groove, small groove, one major groove. These two one turn with 360 angles. Each turn accommodates 10 base pair. In each turn means one minor, one major. The total number of nitrogen bases pair 10. 10 base pair. In 10 base pair, how many nitrogen bases? Hello? Base pair 10 or nitrogen base or nucleotides. How many nucleotides?
Four,
Hello?
four nucleotides, sir.
That is actual type of nitrogen bases 4. I am asking here, in a turn, total like this, 4 type of nitrogen bases, adenine, maybe adenine, guanine, cytosine, whatever it may be. Actual types 4, but total number of nitrogen bases in a one turn, 10 nitrogen bases, 10 pairs, pair 10, but number of nitrogen base,
20.
that's it, 20, 20, right. And the angle separating the two successive nucleotide pairs, 360 degree. 360 to one pair to this total 360, sorry, total 360, total 360, the angle separating two successive nucleotide. So actually in 360-degree angle, how many nitrogen bases? 10 base pairs, 10 nitrogen base pair present. But between angle one nitrogen bases to another nitrogen bases, 36, 36, 36 into 10, how much? Hello? 360. In 360, 10 nitrogen bases. But one nitrogen bases, another nitrogen bases, angle 36 only. Total length. Total length means here, this is one major minor groove. Total turn. Total length 3.5 nanometer or 34 angstroms. 3.4 nanometer or 34 angstroms. Or 34 angstroms. Right? Clear? Hello?
Yes, sir.
Yes, sir.
What is the distance between one nitrogen bases to another nitrogen base? Means, for example, this is what, for example, this is 1 ton, actually 1, 2, 3, 1, 2, 3, 4, so 5, 6, 7, 8. So anyway, remember just 10 nitrogen bases present. So length of the 10 nitrogen bases 34 angstrom. What is the distance between one nitrogen bases to another nitrogen base? Distance. Total 34, but your distance 3.4 angstrom. 3.4 angstroms. One nitrogen bases to another nitrogen bases distance 3.4. Remember. Is it clear? Hello? Watson-Crick model clear? The first of all is Watson
Yes,
and
sir.
Crick model. Hello?
Yes, sir.
Watson and Crick model double helix, right? Discovered by Watson, as I said, Watson and Crick. They awarded 1962 Nobel Prize. So mainly they are arranged at anti-parallel. What is anti-parallel direction? Anti-parallel direction means 5 into 3 end and 3 into 5 end. That is anti-parallel direction, right? Always here the bond formation between adenines, between pyrimidines and purines. Here adenine and thymine 2 hydrogen bond, guanine and cytosine 3 hydrogen bond. So the diameter of the strand 20 angstroms. So in each turn, total 360-degree. The each turn here the consist of two grooves, minor and major. In minor and major groove form a one turn. In one turn, 10 nitrogen base pair present. The length of the one turn 334 angstrom. Between one nitrogen base to another nitrogen base 3.4 angstrom, right? Is it clear? Hello?
Yes, sir.
Any doubt in Watson and Crick model?
No, sir.
Right. DNA structure clear? First, DNA structure, DNA
Yes,
chemical
sir.
structure, and Watson-Crick model also completed. Right? Next. DNA replication. DNA replication. Right. DNA replication, actually three types of DNA replication. Disparative, disparative, conservative, semi-conservative. Simple. What is DNA replication first? What is replication? Replica. DNA replication means one
Another
DNA,
copy.
right? One DNA forms another DNA copy. So this is one DNA strand forms another DNA. This is called replication. It is three types. Disparative. In disparative, DNA strand it is new one and again form old one and again form new one. Right. So this is disparative. In disparative, DNA stands, for example, red color old one and again green synthesized newly and again old one formed and again newly synthesized DNA formed. So this is disparate manner. This is present within the strand. And conservative model, in conservative model, the same. For example, this is DNA strand. So one DNA synthesize newly strand and here newly strand green color. In conservative, after synthesized DNA strand, actually DNA strand two strands. After replication, it becomes four. After synthesized four, two red colors formed a one DNA, two green color, formed a one DNA. So to old form a DNA helix and to new form a DNA helix. This is conservative model. And this first two not accepted, the majority, the most of the scientists accepted semi-conservative model. In semi-conservative model means the same here, just wait, semi-conservative DNA stand, green color newly synthesized DNA, but after formation newly synthesized DNA, here helix formation between one new one and old one, new one and old one. Here also same, old one, new one, right? Clear? Hello?
Sir,
Hello?
once again, explain once again, sir.
First of all, DNA replication definition clear?
Yes, sir.
What is DNA replication? What is replication?
Replica, making another copy, sir,
Right,
from
making another
parent.
copy. Here the making another copy three different ways. One is disparative. In disparative altogether, that means in disparative manner, new one, old one together in a stand. Within a stand, new and old all together in a stand. That is disparative. Conservative
Okay.
means, in conservative means, first of all, this is DNA stand. This is mother DNA, right? This after replication, these two mother DNA formation to new synthesized DNA, right?
Yes, sir.
First two green color, first mother DNA. After replication, after replication means each DNA strand makes another DNA. Each DNA strand after another DNA. How many DNA strands are there? How many strands here? After replication, how many strands?
Two starts, sir.
Is this two or four?
Four.
Four.
Two red color, two green color, right?
Yes, sir.
Right. So here the first of all, here the two green color I consider as a old one. O. Two red color new one. N. After completing of replication, according to some scientists, these two means this new one. New. What are the new color? Red or green?
Red.
Two red spirally coiled form a DNA. And what are the remaining? New or old? Remaining strands.
Old.
Old. Two old together form as a DNA strand. So this is the conservation method of replication. Here only these two old, two new spirally formed, right? Hello?
Yes, sir.
In conservative model, DNA strand after synthesis, two strands are new, two strands are old only. But in semi-conservative model, after replication process same. After replication, after replication means after one DNA strand becomes two. That means total here the two strands becomes four strands, right? Hello?
Yes, sir.
Any confusing?
No, sir.
Tell me if you have any confusing. I will start with first.
No, sir. Semi-conservative only I am having some doubt.
Right. What is semi?
Uh, Half.
Right. So after formation four, here the four means, for example, these two considered as these two red color old, green color new. after replication, after formation 4 strands, the helix formation between one new strand, one old strand. Semi. Here also same one new strand and one old strands. So this method called as a semi-conservative method, right? Most of the
Yes.
scientists accepted semi-conservative method. For example, only through this method, characters are carried forward to one generation to another generation, or one cell to the another cell. For example, I will clearly explain in cell division. First, let me complete in brief here. For example, this is cell. In cell, first, first, two-strand DNA present. After replication, after replication, this two-strand DNA, 4 strands, formation 4 strands. So this is mother cell. A division after cell division, it becomes two daughter cell, right? Hello,
Yes, sir. Yes, sir.
two daughter cells. During cell division, one new strand, one old strands enter into the one cell. Same here also one new, one old enter into the cell. So mother cell DNA 2, here also 2. But here what happened? The characters, half of the characters transfer to the one generation to another generation, one cell to the another cell. It would be continued. It would be continued means, for example, here also two-strand DNA, here two-strand DNA formation into four-strand DNA, sorry, four-strand. Here also same four-strand. When cell division, after formation two daughter cell, same one new strand, one old strands. It would be continue. So this is method called as semi-conservative method. Is it clear?
Uh, yes, sir. Understood, sir.
Right. Right. Now let's clearly discuss about replication process. So concept clear. Replication means one strand we can see here. This is one strand prepared another DNA blue color, and this is another strand prepared another blue color DNA strand. Right. So this is DNA replication. So before entering into this topic, let's discuss about the enzymes. Some enzymes involved in DNA replication process. The enzymes are, just wait, right. In mainly in prokaryotes, three type of enzymes are there. Right. In prokaryotes, three type of enzymes. They are DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. In eukaryotes, there are 5 types of alpha, beta, gamma, delta, epsilon. Are you able to see? Hello?
No, sir.
Are you able to see now?
Yes, sir.
Right. So this is prokaryotic enzyme, only three type: DNA polymerase 1, DNA polymerase 2, and 3. And eukaryote, in place of the three type of polymerase, here the 5 types of DNA polymerase. They are alpha, beta, gamma, delta, epsilon. We will discuss later. First, let me complete about DNA polymerases. Apart from the DNA polymerase enzyme, another important enzymes are, just wait here, the helicase, gyrase, topoisomerase, helicase, gyrase, and topoisomerase. First, let's discuss DNA polymerase 1. This helps in synthesis of DNA 5' to 3' direction. Already you know DNA synthesis means one DNA strand prepare another DNA, right? So for example, here remember one thing, newly synthesized DNA always 5' to 3' direction. Newly synthesized, for example, this is 5' to 3', 3' to 5'. Anti-parallel, right? Is it clear? Hello?
Yes, sir.
Right. For example, this is A stand, this is B stand. Already given one statement, that is newly synthesized DNA strand always 5' to 3'. Remember it. Remember 5' to 3'. Okay. So this is old one 5' 3'. So where is the anti-parallel to newly synthesized DNA? The anti-parallel this direction. Anti-parallel this direction. So this should be 5'. This should be 3'. What about this direction? Hello? In B side. In B side, same. This is 5'. This is 3'. Just show the arrow marks. Show the arrow marks. So in both stand, the direction, in both green color stand, direction 3' to 5' or 5' to 3'. Hello? Arrow
3
mark
into
direction.
5.
Arrow mark directions. Newly synthesized arrow mark directions. Newly synthesized green color direction.
Yes, sir.
Which direction? 3' to 5' or 5' to 3'.
That is 5 into 3.
Right, remember it, 5' to 3'. Always newly synthesized DNA 5' to 3'. So this is synthesized with help of DNA polymerase 1. This is with help of DNA polymerase 1. DNA polymerase 1. And this is called as polymerization reaction. Polymerization reaction. And another important of DNA polymerase, it shows exonuclease activity. Exonuclease activity at the direction of 3' to 5'. Polymerase action. Polymerase action means nothing but synthesizing stand. Synthesizing stand always 5' to 3'. Remember, and exonuclease means here the removing hydrogen bonds and separating the DNA strand. The separated the DNA strand 3' to 5' direction. But newly synthesized DNA 5' to 3' both are opposite. Remember, both are opposite. So one is 5' to 3' direction synthesizing, that is polymerase activity. The polymerase activity of polymerase 1 5' to 3' direction. See, simply polymerase means synthesizing stand. Synthesizing stand. And exonuclease activity means removing stand. The removing the two strands 3' to 5' direction, synthesizing 5' to 3' direction. The major part of the DNA polymerase structures have been called as clino fragment. DNA polymerase 1, another name is clino fragment. So two action. DNA polymerase 1, two action. One is synthesizing DNA, removing DNA. But synthesizing DNA direction 5' to 3'. Exonuclease activity 3' to 5'. Is it clear? Hello?
Yes, sir.
Right. Next one, DNA polymerase 2. Actually, DNA polymerase 2 resemble to the DNA polymerase 1, but it is not involved in replication process. But it can repair. It can repair DNA. It can repair DNA. I will tell you later how the DNA repaired. Next, DNA polymerase 3. This participates in DNA replication and shows synthesized in direction 5' to 3', like a polymerase 1. Polymerase 1 also, but one different is polymerase 1 dual function. Dual function in synthesizing 5' to 3', exonuclease activity 3' to 5'. But in polymerase 2, oh sorry, DNA polymerase 3 only synthesizing one direction 5n to 3n direction, but not exonuclease activity. Okay? Hello?
Yes, sir.
We clearly discussed next class about DNA replication and chromosomes. Clear?
Okay, sir.
Any doubt? Any doubt today class?
No, sir.
Right. Again, we will discuss DNA replication. Okay, in next class. And the chromosome, this is very important. The Watson Crick model, very, very important. And the last class, plasma membrane, nucleus, both are very, very important. Plasma
Yeah.
membrane, in last class, three completed, cell wall completed, plasma membrane completed, nucleus completed. Among those, plasma membrane structure, plasma membrane model, nucleus structure, very important. And today class DNA structure and Watson Crick model. Watson Crick model, very, very important. And DNA replication also. Again we will discuss next class DNA replication and also chromosomes. Right?
Okay,
Hello?
sir.
Any
Okay, sir.
doubt?
No, sir.
Right.
Uh, s-
Ah,
Sir, our paper pattern has changed, sir.
paper pattern changed. Okay, right. Exam pattern, examination.
Yes,
Your
sir.
theory exam.
Yes, theory exam, sir.
Only instead of objectives, they give another
Yeah.
essay type of question.
Yes, sir. It's for 20 marks, sir.
Yes, for 20
So,
marks. Just simple question, way of asking different.
okay. So,
Question
like,
way of
it's written as a knowledge-based test. So,
yes.
it is from our semester 1, 2, 3 also. That question will touch, sir.
No chance. Only the question come from in your course only. In fifth semester course only. Not fourth, third, second.
Okay, sir. Okay.
That is the you're the testing the knowledge only the present existing course only.
Okay.
If
So,
you
if
fifth
we go
semester,
through
the
this,
question comes from fifth semester only,
okay, sir.
right? But it would be lengthy.
Okay, sir. Okay.
For
If we
example,
go through the syllabus only, like perfectly, we can answer that question, sir.
yes, that's it,
Okay.
right?
Thank you, sir. Thank you very much.